The structure of xylem is made up of 4 different types of cells: vessels, tracheids, which are dead, fibres, and xylem parenchyma. Vessels and tracheids are dead cells, and they form a system of tubes through which water can travel. Cells are dead because the lignin has deposited on the cellular cell walls, rendering them impermeable to water and solutes. These cells also provide mechanical strength support to the plant. Water uptakes by the roots. The large quantity of water lost through the stomata of the leaves by the process of transpiration must be replaced with water from the soil. The region of the greatest uptake is the root hair zone, where the surface area of the root is increased by the presence of root hairs. Soil water contains a very weak solution of mineral salts and so has a high water potential. The vacuole root hair cell contains a strong solution of dissolved substances and low water potential. Water passes into the root hair cell down a water potential from high water potential to low water potential by osmosis. Water can travel across the cells of the cortex of the root along 3 pathways. Okay, I have no idea how to pronounce these, so you're just going to have to bear with me. The apoplast, which is through the cell wall, the symplast, which is through the cytoplasm and plasmodesmata, the vacuole pathway, which is vacuole to vacuole. Even though the vacuolar pathway is the fastest, the other 2 are the main pathways. So that's the apoplast and the symplast. The xylem tissue is found in the centre of the root and is surrounded by a single layer of cells called endodermis. Endodermis cells are impregnated with a waxy material called suberin. This forms a distinctive band known as the Casparian strip. The Casparian strip prevents the apoplast pathway, as it is through the cell wall. The only way that water can pass through the endodermis to the xylem is along the symplast pathway, which is through the cytoplasm and plasmodesmata. There is some evidence that salts may then be actively secreted into the vacuolar tissue from the endodermal cells. This makes the water potential in the xylem more negative, as it contains less water and more solutes. This causes water to be drawn in from the endodermis, so promoting the movement of water into the xylem from the cortex. The water potential gradient creates a force known as root pressure. Uptake of minerals. Minerals are taken up by the root hairs by active transport from the solution. Once absorbed, mineral ions move along the apoplast pathway, which is the cell wall. They are carried in solution by the water being pulled up by the plant in the transpiration stream. When minerals reach the endodermis, the Casparian strip prevents any further movement along the cell walls. The ions enter the cytoplasm of the cell wall, from where they diffuse or are actively transported into the xylem. For example, nitrogen enters the plant as nitrate ammonium ions, which diffuse along concentration gradient into the apoplast stream, but enter the symplast by active transport against the concentration gradient. They then flow via the plasmodesmata in the cytoplasmic stream. At the endodermis, ions must be actively taken up to bypass the Casparian band, which allows the plant to selectively take up ions at this point. The movement of water from root to leaves. Water travels in the xylem up through the stem to the leaves, where most of it evaporates from the internal leaf surface and passes out as vapour into the atmosphere. The transpiration of water from leaves draws water across the leaf from the xylem tissue along the same 3 pathways as the root, which is the apoplast, the symplast, and the vacuola. As molecules leave xylem cells in the leaf, they pull up other water molecules. The pulling effect is known as a transpiration pull, and it is possible because of large cohesive forces between water molecules and adhesive forces which exist between water molecules and the hydrophilic lining of the vessels. These 2 forces combine to maintain the column of water in the xylem. The theory of this mechanism by which water moves up the xylem is known as the cohesion tension theory. The capillarity is another force that may contribute to the rise of water in the xylem. Water rises up narrow tubes by the capillary action, but this is more relevant in small plants than in large trees. Transpiration. There is a problem there are many problems that confront terrestrial animals. They have to avoid desiccation and are continually losing water vapour to the atmosphere. 99% of water absorbed by plants can be lost to the atmosphere by evaporation from the leaves. Transpiration is evaporation of water from inside the leaves through the stomata to the atmosphere and gives rise to the transpiration stream. All plants have to balance water uptake and loss. If they lose too much water, they will wilt, become turgor, and then die. Problems with stomata. They need to be open during the day to allow the exchange of gases between tissues of the leaf and the atmosphere. The presence of pores in the leaves mean that valuable water is lost from the plant. 95% of water is lost through the stomata. The other 5% of water is lost through the leaf epidermis, but this is reduced by the waxy cuticle on the leaf surface. The transpiration rate is the rate at which water is lost from plants. It is affected by any factor that increases the water potential gradient between the water vapour in the leaf and the surrounding atmosphere. Factors that affect this are temperature. Increased temperature, increased transpiration rate. A rise in temperature provides additional kinetic energy for the movement of water molecules. This additional energy accelerates the rate of evaporation of water from the walls of the mesophyte cells, and if the stomata are open, it speeds up the rate of diffusion of water vapour into the surrounding atmosphere. Another factor that affects the transpiration rate is humidity. Increased humidity results in decreased transpiration rate. The air inside the leaf is saturated with water vapour, but the humidity of the atmosphere surrounding the leaf varies, with values exceeding 70% rare in Britain. Thus, the water potential gradient between the leaf and the atmosphere is great when there is barely any humidity, and when stomata are open, water diffuses out. The third factor that affects transpiration rate is air movement. Increased air movement results in an increased transpiration rate. This is because transpiration in still air results in the accumulation of saturated air and the Earth's surface. This offers resistance to diffusion of water vapour through the stomata and reduces the rate of transpiration. Okay, so that explanation didn't really make my first bit sense. So, decreased air movement results in decreased transpiration because when there's barely any air movement, all the water particles basically sit on the leaf, and this builds up water outside the leaf so that the water potential outside the leaf is higher than inside the leaf. And the final factor is light intensity. Increased light increases transpiration rate as it controls the stomata opening. When you're thinking of these factors, think of a t-shirt drying outside on the washing line. So if it's more humid, it won't dry as quickly. If it's lighter, it will dry quicker. If its temperature's increased, it will dry quicker, and if there's more air movement, it will dry quicker. So basically, the quicker it will dry, the faster the transpiration rate, if that makes any sense at all. These factors do not work independently. For example, more water is lost on a dry, windy day than a humid, still day. This is because the substomatal chambers has a high water potential, as the walls of the spongy mesophyte is saturated with water. The water evaporates from the walls and moves down a gradient of water potential from the plant to the atmosphere, which has a low percentage humidity. The wind having reduced the thickness of the layer of saturated air at the leaf surface. Mesophytes, Hydrophytes, and Christ, I'm not going to be able to pronounce this. Xerophytes. I'm not really too sure about pronunciation. Right. Plants can be classified on the basis of structure in relation to the prevailing water supply into three groups. Mesophytes. This is plants that have adequate water supplies. Hydrophytes. These are plants that have a lot of water. And Xerophytes. Plants that live in conditions where water is scarce. If a plant loses too much water, the plant wilts and the leaves drop. The leaf surface is reduced and photosynthesis becomes less efficient. Mesophytes. These are most land plants growing in temperate regions are mesophytes, and so are crop plants. Usually the water they lose from transpiration is readily replaced by uptake from the soil, so they do not require any special means of conserving water. Excessive water loss is prevented by the stomata. They flourish in habitats of adequate water supply. They are adapted to grow best in well-drained soils and moderately dry air. Mesophytes need to survive unfavourable conditions during the year, particularly when the ground is frozen. Trees and shrubs shed their leaves before the onset of winter. Aerial parts of many non-woody plants die off as a result of frost or cold winds, but their underground organs survive. Most annual mesophytes, which are plants that flower, produce seed, and die in the same year, survive the winter as dormant leaves. I think that's meant to say seeds, I think. Right. Xerophytes. Show exomorphic adaptations. These plants have adapted to living in conditions of low water availability and have developed modified structures to prevent excessive water loss. They may live in hot, dry desert regions or cold regions where soil is frozen or exposed to windy locations. For example, marram grass, otherwise known as Amorphilia arenaria. Just remember marram grass. These colonise on sand dunes around the coast. Sand dune habitat makes it difficult to survive because of rapid drainage of rainwater, no soil, high wind speeds, salt spray, and lack of shade from the sun. Marram grass shows the following modifications. Rolled leaves. The large thin-walled epidermal cells at the basis of the grooves shrink when they lose too much water from transpiration. This causes the leaf to roll on itself. The effect of reducing the leaf area from which transpiration can occur. Sunken stomata. Stomata is found in the grooves on the inner side of the leaf. They are located in pits or depressions so that water vapour is trapped outside the stomata. It reduces water potential between the leaf and the atmosphere and so reduces the rate of diffusion of water. Hairs. They have stiff and interlocking hairs. These trap water vapour and reduce the water potential gradient. And finally, they have a thick cuticle. There is a waxy covering over the leaf which reduces water loss. The thicker the cuticle, the lower the rate of transpiration. Hydrophytes. These grow submerged or partially submerged in water. For example, the water lily. This is rooted to the mud at the bottom of a pond, but has floating leaves on the surface of the water. Hydrophytes are adapted as follows. The water is supported by a medium, so have little or none lignified support tissue. It's surrounded by water, so there's little need for transport tissue, so xylem is poorly developed. The leaves have little or no cuticle. The stomata are found on the upper surface of the leaves. Stems and leaves have large air spaces, forming a reservoir of oxygen and carbon dioxide. These gases provide a buoyancy to the plant tissue when submerged. Translocation. Structure of the phloem. It is living tissue and consists of several types of cells. The sieve tubes. These are the only components of the phloem obviously adapted for the longitudinal flow of material. It's formed from cells called sieve elements placed end to end. End walls do not break down but are perforated by pores. These areas are known as the sieve plates. Cytoplasmic filaments containing phloem protein extend from one sieve cell to the next through the pores in the sieve plate. They do not possess a nucleus, and during their development, most of the other cell organelles disintegrate. The other type of cell that phloem is made up of is the companion cell. Each sieve tube is closely associated with at least one companion cell. Companion cells have a dense cytoplasm, a large centrally placed nuclei, many mitochondria, and are connected to the sieve tube via the plasmodesmata. Transport in phloem. Experiments suggest that phloem is the tissue concerned with the translocation of organic substances. Several techniques have been used to work this out. Number 1. Ringing experiments. Cylinders of outer bark tissue were removed from the woody stems, and the contents of the phloem above and below the cylinder were later analysed. Radioactive tracing. This is with labelled metabolites using This is a sort of technique with labelled metabolites using aphids. An aphid has a hollow needle-like mouthpart which is inserted into sieve tubes to feed on the sap. To sample the phloem sap, the aphid is anesthetized and the stylet is cut off, leaving it attached to the plant. As the sap is under pressure, it exudes from the very fine tube and can be collected and analysed. And the final technique is radioisotope labelling, where carbon dioxide is labelled with carbon and is supplied to illuminate the plant leaf. The radioactive carbon is fixed in the sugar produced in photosynthesis and is translocated to other parts of the plant and can be traced using autoradiography. The leaf and sink tissues are placed firmly on the photographic film in the dark for 24 hours. When the film is developed, the presence of radioactivity in parts of the tissue shows up the fogging of the negatives. The technique shows that the sugar is transported upwards and downwards direction, since the radioactivity is shown in the aerial parts of the plants as well as the root. An autoradiograph of a transverse section of a stem shows only fogging where phloem was in contact with the film. Theories of translocation. Mass flow hypothesis in 1937. This basically said there is a massive mass flow of sugars from the phloem of the leaf, where there is the highest concentration, the source, to other areas such as growing tissues where there is lower concentration, the sink. Alrighty, that's the end of that one.